When talking about the general structure of an amino acid, in addition to the amine group and the carboxylic group, there's also something known as an R group. And depending on what group you have attached as the R, will let you know what amino acid you have. There's 4 general types of R groups you can have, and it's based on how they treat hydrogen. We're going to break them up individually. First up, acids. There are 2 R groups that are acidic groups, and if it's an acidic R group, that means it's willing to give up a hydrogen. The 2 amino acids that have acidic R groups conveniently end in acid, aspartic acid and glutamic acid. When that hydrogen is gone, it's called glutamate or aspartate, respectively. In addition to acidic R groups, there's also basic R groups. The basic R groups are going to want to accept a hydrogen. To help me remember basic R groups, I use the phrase hoes, ratchets, and Kardashians are basic because the 3 basic amino acids are H, histidine, R, arginine, and K, lysine. So H, R, and K, hoes, ratchets, and Kardashians are basic. And histidine, arginine, and lysine are the 3 basic amino acids. In addition to acidic and basic R groups, there's also polar and nonpolar R groups. The polar R groups are also known as hydrophilic because they're able to create hydrogen bonds with water. So they're attracted to water, hydrophilic. To help me remember the polar R groups, I remember the phrase society and equal is polarized. And all of the consonants in society and equal, there's not an L, are the polar amino acids. S, serine, C, cysteine, T, threonine, Y, tyrosine, N, asparagine, and Q, glutamine are all the polar amino acids. In contrast to the polar amino acids, there's also nonpolar amino acids, which are also called hydrophobic because they are afraid of water. They're not creating polar bonds, so they're not able to form hydrogen bonds with water. They're afraid of water, hydrophobic. Oil is an example of a hydrophobic molecule. If you ever pour oil into water, you'll notice that the oil is going to form clumps. It's not going to easily disperse and dissolve into the water. So to help me remember nonpolar molecules, I remember the phrase, various molecules forming groups linked in water aren't polar. The first letter of each of those words represents one of the nonpolar amino acids. V, valine, M, methionine, F, phenylalanine, G, glycine, L, leucine, I, isoleucine, W, tryptophan, A, alanine, and P, proline are all the nonpolar amino acids. Once the R groups come in, you can create the tertiary structure of the protein, where depending on what interaction you have between 2 R groups, you're going to create a different structure of the molecule. So for example, 2 nonpolar amino acids are going to want to clump towards each other and away from water, whereas 2 polar amino acids are going to want to create a hydrogen bond between each other. And acidic and basic amino acids are going to create ionic bonds between each other. In addition to ionic and polar bonds, as well as hydrophobic interactions being created for the tertiary structure of a protein, there's also disulfide bridges that are able to be created specifically from cysteine molecules. Cysteine is the only amino acid whose sulfur is able to engage in disulfide bridges. And when 2 cysteine molecules have created a disulfide bridge, it's called a cysteine molecule. Disulfide bridges increase the stability of the protein overall. So especially for proteins that have to be secreted or leave the cell, the more disulfide bridges they have, the better it is. So any protein that has to be secreted from the cell tends to have a lot of cysteine molecules and disulfide bridges present. One of those secreted proteins, keratin, which helps grow your hair, the more disulfide bridges you have, the curlier your hair becomes. To help me remember that cysteine helps with extracellular proteins, but also makes your hair curlier, I remember the cysteine cistus. Once R groups within the same peptide chain have interacted, if that peptide chain interacts with another peptide chain, those peptides interacting creates the quaternary structure of the protein. Because all of those R groups are classified based on how they deal with hydrogen and or water, if you change the hydrogen concentration in which a protein exists, aka change the pH, you can also change the actual structure of the protein and whether or not it's going to function. And science is actually getting to the point now where if you know the primary structure of a protein, you can predict what the actual protein is going to look like. Watch the next video to learn more about how pH changes the function of a protein, and you can predict whether or not the protein is going to function by calculating something known as the pI.